So in the previous video, we talked about a transamination reaction. And essentially what we did with that transamination was we took an alanine residue, we took an enzyme, and I'm going to say this enzyme had PLP, or pyridoxal 5' phosphate there, and we also took well, actually that's what we did there, and we generated an alpha keto acid. The specific alpha keto acid that you would generate with alanine would be pyruvate. And we also produced, so alpha keto acid, I'm going to put pyruvate in parentheses. We also generated our enzyme PMP. Okay, so our nitrogen from here ends up here. Sorry, our nitrogen from alanine ends up on the PLP, which is now PMP. Okay, so this right here is something that we have to deal with because we haven't really lost the nitrogen. All that we've done is we've transferred it. Okay, so now basically the second part of this is enzyme dash PMP plus another or a different alpha keto acid. In this case, our alpha keto acid is going to be alpha keto glutarate. What we're going to generate here is glutamate. And we will also generate our enzyme PLP because we've transferred the phosphate group off of our alpha keto acid and onto our alpha keto. Sorry, we transferred our nitrogen from enzyme PMP to our alpha keto acid or alpha keto glutarate, and so that gives us our glutamate, and we returned our enzyme PLP to its original state. So we could begin doing this transamination reaction again. Now, this glutamate has the nitrogen that we actually want to get rid of. And in order to do that, in the liver, we would use an enzyme known as glutamate dehydrogenase. Now that glutamate dehydrogenase will help us basically regenerate alpha ketoglutarate and it will also help us basically release NH4+, so our ammonium. Okay, so this is in a nutshell kind of what we're wanting to do. What we want to do is take something like alanine and take the nitrogen off of that and ultimately get rid of it. But there's a little bit of an involved process with that. The alanine has to donate that nitrogen, or that NH2 as it would be, onto our PLP, which becomes PMP. The PMP then donates the nitrogen it donates it to our alpha keto acid to become glutamate. That glutamate then donates that nitrogen to produce alpha ketoglutarate, which what can it do? It can jump back up here, and our NH4 can ultimately be excreted. We'll continue with this in just a second. So what we were just talking about on the previous slide, or the previous discussion, was this transaminase. Now, to put all this into perspective, this right here is a liver cell. And within that liver cell, we've got our mitochondria, and we got our cytosolic space. So the transamination reaction is taking place in the cytosolic space. Now, from that transamination, we're generating our alpha keto acid, which is pyruvate, and we're also generating glutamate. Now, glutamate dehydrogenase is an enzyme that's going to take glutamate and regenerate another alpha keto acid, or alpha ketoglutarate, but it's also going to release our NH3 that we're ultimately wanting to get rid of. Now, the enzyme that is going to do this reaction, glutamate dehydrogenase takes glutamate, generates NADPH, generates NH3, and produces alpha ketoglutarate. Now, this NH3 that we're aiming to ultimately release and get rid of, well, that is going to be used in conjunction with this HCO3 and 2 ATP molecules by the enzyme carbamoyl phosphate synthetase. That enzyme is manufacturing or producing this carbamoyl phosphate. So we have our carbons there. We have our nitrogens, which originally came from our alanine, and we also have this phosphate group here that came from one of our ATP molecules. Okay, now this enzyme is also going to be producing 2 ADP molecules and 1 inorganic phosphate. So these 2 ATP molecules that we're contributing to this, one of them, well, transfers a phosphate group onto to make our carbamoyl phosphate. Another inorganic phosphate is simply released. So the other side of this reaction that we were talking about, or the other product that we had, was our pyruvate molecule. So the pyruvate that evolved from our transamination reaction. One fate of this is that it can go on to ultimately provide a source of glucose through gluconeogenesis, because again, we are in the liver, so the liver is capable of doing that gluconeogenesis. Sorry for that line there. Alternatively, pyruvate can be funneled into the mitochondria and ultimately go on to make aspartate. And the pathway that we're about to go into, the relevance of that, or the production of that aspartate, is particularly valuable. But more than anything, what I want to focus on is the generation of this carbamoyl phosphate, because this molecule is the molecule that is going to enter what is a cycle known as the urea cycle. Now, the cool thing about this is, and it really kind of speaks to, I guess, intellect and intelligence and, well, one person's hard work, or not just one person's hard work, but this pathway, this urea cycle, is also known as the Krebs-Henseleit cycle. So this is the same Krebs responsible for the tricarboxylic acid, or the Krebs cycle. Alright, well, we're going to jump into the Krebs cycle in the next video, but I wanted to show you guys kind of, well, what we needed to do to get our molecule that would ultimately enter the Krebs cycle. So, I look forward to you in the next video. Have a good one.